and digest more proteins that are now a bigger part of our diet. So I saw a number of disconcerted head shakes when we were talking about this. What is it that you don't like about this? What is it that's confusing?
The steps. I just think the little
Everything, the whole thing.
little things about it, like it connects.
Okay, so let's come back to that, Mac. Adam?
So this is the process of what again?
How to make a
That's a
protein.
wonderful question. What are we doing here?
Making a
It
protein.
leads to the digestive
We're
tract.
creating a protein for
The digestive
digestion. Okay, so I think if you guys try and like tackle this wholesale, big picture all at once, it's going to be overwhelming. But if you think about the constituent parts, it's not that bad, right? I mean, do we know what the nucleus does? What does the nucleus do? Controls cellular activity. It holds on to DNA, right?
Yeah.
And DNA is nothing more than a blueprint for a protein. So if we want to make more proteins, we need to get a message from the nucleus to the actual things that make the proteins. And what actually makes proteins? Ribosomes, right? So that message is just an RNA copy of our DNA, and that goes and finds a ribosome. One of the tricky parts is, where is the ribosome located? Let's say we wanted to make a protein that's going to, we want to make a kinesin that's going to be carrying vesicles inside the cell. Where, what's that?
Goes in the cytosol.
Yeah, the ribosome is in the cytosol, right? Because that kinesin is staying inside the cell. Exactly right. So if the protein is to stay inside the cell, the ribosomes are located in the cytosol. If that protein is going to be secreted from the cell, it's located in the rough endoplasmic reticulum. Is that an okay idea? Okay, so if we want, oh yeah, go ahead. Kira.
So, we used to keep talking about kinesin, so like, when would you ever use the dynein?
Dynein?
Yeah.
Oh, that is a wonderful question. Can I come back to that question
Yeah.
after I'm done explaining this stuff? Okay. So if we're making a protein in the endoplasmic reticulum, which is a part of the endomembrane system, we need to get that from the ER to the Golgi, right? And ER is just membrane, so we can make a vesicle membrane, a membrane vesicle, out of the ER, just butt it off and send it along from the ER to the Golgi. And we know that there are microtubules, there are highways along which vesicles move, right? So it's just walking along a microtubule from point A to point B. The structure that completes a protein is the, see, this is the part where I'm losing attention again. Right, it's the Golgi, right? The Golgi is what actually completes the protein. And then we just need to get that completed protein from the Golgi to the surface of the cell, and again, that's just microtubules. They accomplish within cellular movement. Yeah, Adam.
So is there like tons of those, like
Tons.
the whole cell like it's just covered?
Tons. Like, you should look up images of the cytoskeleton. And it will have different colored fibers running to different parts, running throughout the cell. And there will be microtubules and microfilaments, and they're just everywhere.
Vesicles attach themselves to that.
They do. They do. With either a dynein or a kinesin. And so Kira's question was, when do we use dyneins? And that is a wonderful question. So, in mitosis, what happens is we get two centrioles, and they've been replicated, right? We need to have two centrioles because each daughter cell needs to have one. So we have two centrioles inside the cell, and we have chromosomes that are just kind of floating around like this. And eventually what happens is microtubules from each side grow out and attach to all of the chromosomes, so that ultimately we get chromosome alignment like this at the center of the cell. When that happens and those microtubules are still attached, I'm not going to draw the centrioles just for the sake of expedience. when that happens, and this is the origin of those microtubules, right? These sides of the chromosomes are going to be pulled apart, and they're going to start migrating this direction. They're actually going to have proteins carrying them from the center of the cell to the origin, and that's when diamines, one of the uses of diamines. So they're going to carry part of that chromosome from here back to the origin. Other questions? Okay. So, oh yeah, Adam, go
What
ahead.
is that process called again?
Mitosis? So I'm going to take a picture of this really quickly for my internet students who I know aren't going to be able to see that very well on the board. Okay. Other questions about that? I'll leave that up until the end of class. However, I will say that you guys need to know these structures in that kind of detail for the exam. Like that's a really, really good written item. All right?
Can you wait on that one?
On this one?
Yeah.
Yeah, for sure.
Is that one gonna be on the exam?
This one? No, not yet.
Not yet, okay.
Okay, so now we need to talk a little bit about the outside of the cell. And we're going to talk about plant cells and animal cells a little bit differently.
Thank you.
Yeah, you bet. So all cells have a plasma membrane. But plant cells have an additional structure outside the plasma membrane. And that's called the cell wall. And you'll remember when we were talking about biologically important molecules, we talked about cellulose, which is the structural polysaccharide that makes up the plant cell walls. So what happens with plant cells, and you guys saw this a little bit yesterday when we looked at Belodia, all plant cells have a plasma membrane inside them. And that's what this is. And those plasma membranes have proteins incorporated into them, and those proteins actually manufacture cellulose. And they secrete what's called a primary cell wall. And it's generally located first just adjacent to the plasma membrane. Now, all plant cells have a primary cell wall. And it's cool because it's a little bit rigid, but it allows some bend as well. And so think about like a blade of grass. Grass blades, those cells all have primary cell walls. So you can flick a blade of grass and it can flop over and hit the ground, and then it'll just pop back up. So they're a little bit stringy. And then sometimes what happens, if you really want that cell to maintain its shape, is you secrete what's called a secondary cell wall. So if you secrete a secondary cell wall, and I'll just write this is primary. And this is plasma membrane. You already have a primary cell wall, and it's the plasma membrane that's secreting or manufacturing the cellulose. So the plasma membrane secretes a much more rigid structure called a secondary cell wall. That is in between the plasma membrane and the primary cell wall. So if you were to list, if a plant cell, and not all plant cells have secondary cell walls, things like woody vegetation like the trees behind us, those have secondary cell walls because you don't want that trunk to be flopping back and forth all that much. That secondary cell wall is super thick, super rigid, and and it maintains the shape of the cell. If you were to order the layers of a plant cell surface from inside to out that has a secondary cell wall, it would go plasma membrane, secondary cell wall, primary cell wall. And then there's some gooey stuff that's located in between primary cell walls, and that's called the middle lamella. It's just like glue that holds adjacent cells together. So we have a cell wall, or excuse me, we have a cell here and a cell here. This big stuff in the middle, that's going to be secondary cell wall. This little stuff right here, that's going to be primary cell wall. And then in between them, that's middle lamella, primary cell wall, secondary cell wall, plasma membrane. So if it's kind of a sticky substance, what would you assume?
Sap.
Yeah, it's very much like sap. And sap is sticky because why? It has a bunch of one type of molecule in it. If you guys are eating honey and you get it on your face gets sticky, right?
Sure.
It has a bunch of sugar in it. Plants are really, really good at making sugars. So that middle lamella has a very high saccharide concentration, very much like the sap does that oozes from trees. Okay. Questions about that? All right. So let's talk about the extracellular matrix really quickly in animal cells. So our cell, we're multicellular, and our cells need to be connected to each other. So there is a matrix of stuff which largely consists of proteins, some sugars, and some fats. So there's a gooey substance that kind of runs in between our cells, kind of like the middle lamella. And then we need to attach our cells to that gooey substance. Now the gooey substance is mostly protein. It's mostly collagen and elastin. Elastin particularly is called a glycoprotein. It's a protein that's attached to sugars. So that's the gooey stuff. And then we have little proteins that extend up from the surface of our cell called fibronectins that connect our cells to that extracellular matrix. And that holds adjacent cells together. But it's more important than that, you guys. The extracellular matrix also becomes a medium of intercellular communication. Cells communicate with each other through that extracellular matrix. Sometimes it's a very physical type of communication. One cell can physically pull on another cell, and that will convey some bit of information. Generally, when cells are damaged, it'll be that type of physical communication. One cell will pull on another and say, hey, I'm in trouble, and then that other cell can do something about it. Either start going through the cell cycle of mitosis or initiate some sort of like digestive process where the adjacent cell can be consumed. And this is what the extracellular matrix looks like. So this is our plasma membrane. And we have these proteins, right, that are located across the plasma membrane. In between adjacent cells, we have things like elastin and collagen. And you'll notice that the elastin right here is attached to these really branchy, feathery-looking things. Those feathery-looking things are sugars. So this is a glycoprotein. Glyco referring to sugar, protein, obviously, protein. And then collagen, which is mostly proteinaceous. So that's the stuff that's running in between cells. And then coming off of these integral proteins embedded in our plasma membrane, there are these strings called fibronectin. And the fibronectin becomes connected to that extracellular matrix and is the medium that, again, allows cells to communicate and stay anchored. Okay, questions about the extracellular matrix? We will start with osmosis and diffusion and active transport when we come back on Monday. And we'll probably start chapter 5. So have those printed out. Yeah, Linda.
I have just a broad question. So
Okay.
I'm reading this book I picked up from the library that says why sugar is deadly
Okay.
in the body. If our bodies, the protein with the sugar and everything is all for our body, then why is it so harmful for our bodies to ingest more sugar?
So sugar by itself isn't harmful, right? I mean, it's necessary. That's how we store energy,
Okay.
partially, as glycogen. It's the type of sugar and the amount of sugar that we're consuming.
Okay.
So you can compare sugars and their healthfulness by something called the glycemic index, and that's how sugars are absorbed by our bodies. Sucrose, which is table sugar, has a very high glycemic index. Our bodies absorb it and store it really, really easily. There are other sweeteners that are largely sugar, like honey and like agave nectar and things like that, that have much lower glycemic index that aren't nearly as bad for our bodies. So it's more type and quantity than it is sugars themselves.
That makes sense. Thank you,
Good
I was
question,
just
though.
curious.
Yeah. Other questions? Okay, then I will see you guys back here on Monday. Please don't forget to take the quiz.